Okay, good morning everybody. Are we ready? Right, let's get started. I think we talked about some part of gluconeogenesis, right? In last class, and we mentioned that the precursors, some of the amino acids, are also the precursors that can be used for gluconeogenesis, and the purpose of the gluconeogenesis is to make glucose during the fasting, right? In order to maintain the blood glucose level, and we also mentioned that the metabolic pathway gluconeogenesis that involve 4 key reactions, right? 2 key reactions are in the mitochondria, and the other 2 in the cytosol. And we also mentioned depend on the precursors, which precursors that our body use, and different precursors can go through the different pathways, right? Remember, some of the precursors can involve the 4 key reactions, and some of the precursors only involve the 3 reactions or even 2 reactions, right? Depends on the precursors. And I think last time we stopped here, right? The glutamate, amino acids can be used as the precursors for gluconeogenesis, but the pathway that we mentioned only involves the 3 key reactions, right? 1 reactions in the cytosol, 1 key reaction in the mitochondria, and other 2 reactions in the cytosol. I don't know why. And also we mentioned that TCA cycles is also involved in the gluconeogenesis when we use the glutamate as the precursors, right? Remember this? So, thank you. Right? And glutamate, converted to alpha-ketoglutarate that enter the TCA cycles and goes through the several reactions in the TCA cycles, and alpha-ketoglutarate eventually converted to oxaloacetate, and this oxaloacetate then directly converted to the phosphoenolpyruvate catalyzed by this enzyme, so PEPCK, phosphoenolpyruvate carboxykinase, and bypass this key reactions, right? So this gives us a little bit more details when we use the glutamate as a precursor for gluconeogenesis. First of all, it's converted to the alpha-ketoglutarate, and then through the several reactions in the TCA cycles, alpha-ketoglutarate being eventually converted to the oxaloacetate, and oxaloacetate then leaves the TCA cycles and then go to the rest of the gluconeogenic pathways for glucose, or synthesis of glucose, right? Conversion of glucose. And I just wanted to mention this again, the TCA cycles is very important, that involve the three macronutrients metabolism, and it's also the key connection, right? For example, when we consume too much glucose, and in addition to just using for energy through the glycolysis for complete oxidation, and excess glucose can also be converted to fat, right? So this is actually the important link. So remember that the glucose can be metabolized through the glycolytic pathway to pyruvate, and pyruvate enter TCA cycle. Enter the mitochondria and convert it to the acetyl-CoA. Remember this reaction, right? And acetyl-CoA is the key molecule that enter the TCA cycles. So for the first reaction, the TCA cycles, acetyl-CoA together with the oxaloacetate, then condense to the citrate. So citrate is also very important. It's the first product, right, in the TCA cycles. It's converted from acetyl-CoA and oxaloacetate, and citrate then leave the TCA cycles and go to the lipogenesis pathways for synthesis of fatty acids, which we're going to talk about later. But just wanted to remind you that kind of connection, carbohydrate metabolism, between carbohydrate metabolism and lipid metabolism, right? So how the excess of glucose, carbohydrate that we consume can be converted to the fat. This kind of is the key point that connected this metabolic, right? Metabolic conversion. And also amino acids can be converted to glucose, right? And this again, the link, right, in the TCA cycles. In addition to glutamate and the other amino acids, any type of amino acid that can be converted to succinyl-CoA can also be used for gluconeogenesis, similar pathway, right? Any questions? All right. So, and the last one, we're going to talk about the glycerol. So glycerol is also can also be used as a precursor, right, for gluconeogenesis, and glycerol only involves the two key reactions in the cytosol, right? Because this is sort of a pathway that for gluconeogenesis. Glycerol as the initial molecules. So glycerol can be phosphorylated to glycerol 3-phosphate by glycerol kinase. So glycerol kinase is very highly expressed or present in the liver. So that's why the liver can pick up glycerol, which initially from the triglyceride breakdown, right? So triglyceride breakdown in the adipose tissue during the fasting, right? We need to utilize the triglyceride as the energy. So triglyceride can be break down into glycerol and the 3 fatty acid molecules. So fatty acids can be picked up by other tissues, utilized as energy, for example, in the muscle and liver as well. The glycerol can also be picked up by the liver and converted to glucose, right? So gluconeogenesis occurs again during the fasting, and glycerol initially is coming from the fat tissue, right, adipose tissue. So the glycerol 3-phosphate being converted to, kind of backwards, right, the glycolytic pathway to dihydroxyacetone phosphate, and this eventually, converted fructose 1,6 phosphate and fructose 6-phosphate, glucose 6-phosphate, and then eventually glucose. So these are the two key reactions in the gluconeogenesis that involved in the gluconeogenesis by glycerol. Right? And energy utilization. So for the synthesis of glucose, we need energy, right, to do that. So 6 molecules of ATPs usually are utilized to produce 1 molecule of glucose from 2 molecules of pyruvate. And in the liver, this energy required for the de novo synthesis of glucose or gluconeogenesis, same thing, right? Usually comes from fatty acids oxidation or partial amino acids oxidation, right? So energy, 6 ATPs required for making 1 molecule of glucose, and this energy, 6 ATPs, basically from fatty acid oxidation or amino acids oxidation, right? So, and this slide just put kind of, together, just remind you that the 2 important cycles for gluconeogenesis, Cori cycles and alanine cycles, right? And these cycles involved 2, actually more than 2, 3, right? Tissues, cells, the liver, muscle, and the red blood cells, right? So for Cori cycles, we know that the lactate basically is released from muscle or from red blood cells, right? I should put the arrow here. Then lactate can be uptaken by the liver, right, and for making glucose. So, and this is the Cori cycles. Again, involve the muscle, red blood cells, and the liver for making glucose, and the precursors for gluconeogenesis in the Cori cycles is lactate, right? And alanine cycles involve the 2 tissues, liver and muscle. So alanine is released from the muscle during the fasting, because of protein some part of protein can be also break down for energy use during the fasting. So alanine will be released from the muscle and picked up by the liver for making glucose. And glucose is very important. It's made from the liver during the fasting to provide energy for brain and red blood cells, right? Again, red blood cells only utilize glucose for energy, cannot utilize fatty acids and other energy fuels such as ketone bodies. The reason is why red blood cells cannot utilize other energy fuels. Sorry? Yes, no mitochondria, right? We're going to talk about later. Fatty acids, when we utilize fatty acids or ketone bodies, we need to break down this molecules, oxidize fatty acid. That all reactions occurs in the mitochondria, right? So if we don't have a mitochondria, the red blood cell doesn't have a mitochondria, so cannot oxidize fatty acids and ketone bodies. So only utilize glucose for glycolysis, right? And anaerobic glycolysis, right? And produces lactate and only 2 ATPs, right? And brain is able to utilize the other energy fuels like ketone bodies, but glucose is a primary fuel, right? They utilize glucose. So our body try to make a glucose and primarily to supply, brain for energy production. Only when we undergo very long-term starvation then we really have a very limited glucose available for brain. Then brain start to utilize alternative fuels, which is ketone bodies, right? We're going to talk later, but not fatty acids. Brain cannot utilize fatty acids for energy. The reason is fatty acids molecules are relatively larger, right? Cannot pass through the blood-brain barrier, right? Okay, so let's do something interesting, clinical correlations. Remember we talk about key reactions, right, in gluconeogenesis and key enzymes. One of the key enzymes called PEPCK is a phosphoenol pyruvate carboxykinase, right? PEPCK is one of the key enzymes that catalyze reaction in which part of the cells, remember? In which part of cells? Mitochondria, right? So it's one of the key enzymes that catalyze reaction in the mitochondria, PEPCK. So if patient have a mutation or whatever reason that have a lower PEPCK production, what will you expect would happen to the blood glucose control during the fasting, let's say? If the patient doesn't have enough PEPCK, what will happen to their blood glucose level during the fasting? Increase, right? They're not able to maintain their blood glucose levels because, gluconeogenesis is a very important activity that makes glucose to maintain the blood glucose level, right? So if patient is not able to do the gluconeogenesis, then they were not able to maintain the blood glucose level. So blood glucose level will go low during the fasting. So the patient will probably suffer from so-called hyperglycemia. They have a lower blood glucose level, right? And what happens to blood lactate levels? Increase. Why is increase?
Because it's not sugar.
Okay, let's see whether it's increase or not. Yes, increase, right? So because you have our body the patient had a lower blood glucose level, right? You try to spare glucose. So a lot of the tissues will not go complete oxidized glucose, will not do the aerobic glycolysis. So a lot of tissue which only do the anaerobic glycolysis, just stop at the partial oxidation glucose glycolysis, right? So then again the glucose will not, in a lot of tissue, will not completely oxidize for energy. So then will generate a lot of lactate. Does that make sense? Right? Because the lactate is the precursors of gluconeogenesis. Although the patient may not be able to do it, but just because sort of a signal, from our body tells us, okay, we have a lower glucose level. We need to do the gluconeogenesis, so we need precursors, right? So a lot of precursors. So a lot of tissue will save the glucose, the carbon skeleton, and try to provide more substrate for gluconeogenesis. Although our body can't do it, right, without enough PEPCK. So this patient will have high levels of lactate, And what happens to glycogen store? Up or low? High or low? Glycogen store? Low, right? Use up, right. So because glycogen overused, I would say, right, in the patient, so try to provide more glucose from the different source, right, from the glycogen. So glycogen will be completed, will be break down to release glucose, to try to compensate the defects, right, in the gluconeogenesis. Okay, so all right, so next case. So how about the defects in glucose 6-phosphatase? What happened to the blood glucose levels during the fasting right? I mean, fat condition, patient will be okay, right? You can provide glucose from food, so it should be fine. But during the fasting, what happened to blood glucose level if we don't have enough glucose 6-phosphatase? Again, this is also the key enzyme, right, for gluconeogenesis in the cytosol. This is actually the last step of gluconeogenesis in the liver or kidney, right? Blood glucose level during the fasting, lower, right? So similar, have the patient will have hypoglycemia, will experience hypoglycemia during the fasting. And what happens to blood lactate levels? Also similar, right? High. So also have high lactate levels during the fasting in this patient. And what about the glycogen stores? What do you expect? Glycogen stores in this patient with low glucose 6-phosphatase activity? How many of you think it's low depletion? Okay. And how many of you think it's increase accumulated? Oh, about the same. Well, actually a little bit more increase. So let's see. Glycogen accumulation. Does that make sense? Right? Because you cannot break down completely, the glycogen to release glucose. And you have lots of glucose 6-phosphate substrate accumulated, right? And this substrate is very strong inhibitors of the glycogen breakdown enzymes, right? So glycogen cannot be break down and then accumulated in the liver. So try to remember, these two cases and have a different consequence in terms of glycogen stores, right? Okay. Yeah, this just give you some, idea that this enzyme is a place that enzyme does, right, to convert a glucose 6-phosphate to glucose. Okay, so gluconeogenesis in kidney. So kidney can also do the gluconeogenesis, right? So usually contribute 10% of gluconeogenesis during the fasting. And as the fasting prolongs, going the longer time that the kidney make more contribute more glucose, right? And so for example, after 24 hours of the starvation, that 50% of glucose that generated from gluconeogenesis from kidneys, right? So kidney, so compared to liver, of course, is the most important tissue that make glucose through gluconeogenesis, but during the prolonged fasting, the kidneys also become important. So can account for 50% of glucose from gluconeogenesis. And precursors mostly are the glucogenic amino acids, right? So kidney mostly utilize the amino acids as a precursor for gluconeogenesis, which is different from the liver, right? Liver can utilize lactate, can utilize alanine, and glycerol as the precursors, right? And kidney mostly utilize amino acids as the precursor for gluconeogenesis, and then involve, of course, the TC cycles, right, for gluconeogenesis. And some concept that we're going to talk about next couple of slides is anaerobic and catalytic reactions, right? So this just kind of concept, involve the TC cycles for gluconeogenesis. Okay, so let's talk about pyrolysis and catalysis. So these are the process that the two process that go the opposite direction, that involve the reactions during the TC cycles. So during the fat or fasting conditions, the TC cycle reaction TC cycles become very active, right? So the substrate or intermediates in the TC cycle has to be maintained the certain concentration of certain levels that in order to keep TC cycle running. So if somehow the TC cycles intermediates is too accumulated or depleted, then will will affect the TC cycle function, right? So in order to maintain the TC cycle running or the functioning, so there are two processes called anaerobic or catalysis that can either adding the more substrate to the TC cycle to replenish the TC cycle intermediates, or can remove some intermediates if the intermediates accumulate, right? So these two processes that is very important in order to maintain, again the TC cycle functioning. So anaerobic means that we adding some 4 or 5 carbon intermediates into the TC cycle. So this process is called anaerobic, right? And catalysis, that means remove the intermediates from the TC cycles. So this process is called catalysis. See, for example, here, let me just put all the circle here. So if so highlight in the blue with all the process, you can see we're adding actually the intermediate to the TC cycles, right? For example, the glutamine can be converted to alpha ketoglutarate. So this reaction we can increase the alpha ketoglutarate in the TC cycle, right? And similarly, for this reaction, the valine or isoleucine or methionine, and this amino acid can also be converted to succinyl-CoA, right? So adding succinyl-CoA to the TC cycles, and phenylalanine or tyrosine also can add fumarate to the TC cycles, and similar for the aspartate or pyruvate, right, converted to the acetate. So all these reactions or processes are considered anaerobic, right, because they can add intermediates into the TC cycles. And the red highlighted two process, for example, the citrate, right, is synthesized in the TC cycles, can leave the TC cycles, so for the lipogenesis, right? So during the lipogenesis, in the fat condition, when citrate synthesized in the TC cycle can be moved away from the TC cycle and utilized for fatty acid synthesis, right? So this, and similar for the oxaloacetate that can be made in the TC cycle and can also be utilized for gluconeogenesis, right? And to take it away from the TC cycle. So these two processes we consider as a catalytic process, right? Now let's talk about the role of TC cycle, the gluconeogenesis. I think we did, right? I just wanted to remind you again that glutamate can be utilized for gluconeogenesis, converted alpha-ketoglutarate, and then converted to succinate, fumarate, malate, and to the oxaloacetate. And this process is the anaerobic process, right? And then oxaloacetate, once it's made, and then can be used for gluconeogenesis and leave the TC cycle. So this process is the catalytic process, right? So TC cycles, the gluconeogenesis from the glutamate involves both catalytic process and anaerobic process, right? Does that make sense? So, and also in the glutamine metabolism in the small intestine that also involve TC cycles and anaerobic process and catalytic process. And remember that we mentioned the small intestine utilize 80% of energy that utilized by small intestines come from amino acids, particularly glutamine, and aspartate, right? So glutamine can be utilized as energy in the small intestine, and glutamine, first of all, being converted to alpha-ketoglutarate, right, at the intermediate to the TC cycle. So this is an anaerobic process, right? And once it's alpha-ketoglutarate being converted to malate, and the malate leaves the TC cycles, and this is again the catalytic process, right? And then malate being converted to oxaloacetate and converted to PEP, phosphate enolpyruvate, and then this PEP then converted back to pyruvate and then enter the TC cycle then for complete oxidation, right? So sometimes our body just does this. I don't know why not directly to go to the TC cycles, right? So maybe some kind of regulated mechanism, for example, during this enzyme activity could be very high in the small intestine, right? So then can convert it, to catalyze this process and to the pyruvate and then enter the TC cycles. Okay, so the other process is called glyconeogenesis. Glyconeogenesis is the process that the glycerol released from the from the adipose tissue can be recycled, right? And for the triglyceride synthesis. So, for example, let me see. Sorry, this is not what I'm trying to say. So, glyconeogenesis means then remember when we synthesize triglycerides, we need a glycerol backbone, right? So glycerol backbone, I think we mentioned before, in the adipose tissue, for glucose, for glycolysis, right? So glucose can metabolize and to generate the glycerol phosphate, right? So this is sort of the pathways. The pyruvate that can be metabolized from glucose or amino acids, both, and then enter TC cycles, right? And utilize TC cycles through the anaprosis and cataprosis process, and for making glycerol phosphate. And this is the important glycerol backbone for synthesis of triglyceride, together with three free fatty acid molecules, right? So again, the glyceroneogenesis pathways involve the cataprosis and anaprosis and TC cycles. The main purpose of glyceroneogenesis is to convert the glucose or amino acids to the glycerol backbone, glycerol 3-phosphate for triglyceride synthesis, right? That makes sense? Okay. All right, so now it's the last not last pathway, right? Third pathway that we're going to talk about, the pentose phosphate pathway, right? So this pathway is relatively simple, but what you need to remember is that substrate for this pathway is glucose 6-phosphate. Glucose 6-phosphate is the substrate for pentose phosphate pathway, right? And goes through the many steps and several steps. And the purpose of a pentose phosphate pathway is to generate the ribose 5-phosphate, so five sugar phosphate, right? And the second thing is that during this pentose phosphate pathway, NADPH, not NDH, right? NADPH molecules are generated, and these NADPH molecules are important and can be used for fatty acid synthesis, right? And then later we're going to talk about it. So just, I think that's enough. You don't need to more. So again, this is pathway is a secondary pathway of glucose oxidation, and this pathway provides, reducing equivalent NADH and NADPH. And again, the NADPH is important for the fatty acid synthesis, and the purpose is the production of 5-carbon sugar phosphates, right? Ribose 5-phosphate and oxidose 5-phosphate, and this reaction takes place in the cytosol. And this 5-carbon sugar phosphate is important for the biosynthesis of nucleotide or nucleic acid like ATPs, DNA, RNA, FAD, NAD, or ADP. This nucleic acids or nucleotides are synthesized from 5-carbon sugar phosphate, right? Again, I already mentioned the NADPH is required for synthesis of fatty acids, cholesterol, and sterol. And in the red blood cells, NADPH is also important for maintaining the integrity of a red blood cell membrane, right? Okay, now something I think is very important. We're going to talk about the regulation of the glucose metabolic pathways. Right? So here we just try to focusing on the two main pathways of glucose, glycolysis and gluconeogenesis. Right? And in terms of regulation, think of glucose transporters, substrate cycles, and hormones that are all involved in regulation of glycolysis and gluconeogenic activities. Right? We know that glycolysis mostly occurs in the fat condition, right? And gluconeogenesis occurs in the fasting. So how our body can switch from oxidized glucose and to making glucose during the different physiological conditions. So multiple things are involved in terms of regulation. Again, the glucose transporters and substrate cycles means the substrate that's generated during the pathways that can also self-regulate the rate of the pathway or activities, and hormones. I just want you guys to focusing on the major hormones like insulin and glucagon, epinephrine, right? And the primary the targets of the hormonal regulation are enzymes, right? The key enzyme. Remember why I ask you to remember the key enzyme in each reactions, because when we regulate the enzyme activities, then key enzyme activities, then we can control the whole pathway activities, right? So transporters, we mentioned that 5 different transporters, right? You probably wanted to keep in mind, like GLU1, GLU2, I would like you guys to focus on GLU2, GLU4, GLU5, right? And GLU2, the mainly is in the liver and pancreas, right? Beta cells, and GLU4, the skeletal muscle, cardiac muscle, and adipose tissue. So GLU2 basically is the glucose dependent, right? Remember, and GLU4 is the insulin dependent, and GLU5 basically is the small intestine usually transport fructose, right? And the new concept I wanted to bring in that the affinity for glucose and Km, right? To these two concepts maybe help us to better understand why different glucose transporter can be regulated differently. Some regulate by glucose level, and other regulate by insulin. So mechanics contents Km. Have you guys learned this before, right? Km. So Km is usually defined as the substrate concentration at the half maximum velocity, right? So remember, for the enzymatic reactions, substrate, enzymes, and products, right? These are 3 things we need to remember. So when the enzyme definitely is the most important control, right? If we increase the enzyme concentration, that definitely will speed up the reaction. But it's also depend on the substrate, right? Substrate. If we don't have enough substrate, even you have a higher concentration of enzyme, that reaction cannot be speed up further. So substrate and enzyme are the 2 important the factors that affect the velocity of the reaction, right? So, and this Km means if we keep the enzyme concentration constant, keep enzyme concentration same, and if we increase the substrate, then we're able to increase the speed, right, of reaction. But this increase can also have a plateau. I mean, saturate, right? If once you add a substrate and reactions can reach the maximum, then even you add more substrate, then reaction will not increase anymore, right? So this is sort of a concept. So the Km means how much substrate concentration is required for reach the half the maximum of the reaction, right? So in other words, that this Km is substrate concentration depend on the affinity, the enzyme affinity for the substrate. So if enzyme affinity for substrate is high, then you don't need much high concentration of the substrate to reach that levels of reaction, velocity, right? So if the affinity of the enzyme for the substrate is low, then you will need more substrate in order to reach the similar levels of reaction. Does that make sense? All right. So affinity and the Km actually is opposite. So if, for example, if enzyme have a high affinity for substrate and should have a lower Km, right? And if affinity enzyme have a lower affinity for substrate, and the Km should be high. Does that make sense? Okay. So and the glucose transporters, we consider like enzyme, right? And the glucose as a substrate. So a different glucose transporters have a different affinity or Km for glucose. So for example, group 1 and group 3 usually have a lower Km or high affinity for glucose. So the group 1 and group 3 basically is kind of more active even when we have a lower blood glucose levels. So group 1 and group 3 basically transport glucose at like a basal level, right? Even you have a lower, blood glucose level, for example, under fasting condition, and group 1 and group 3 can be active to transport the glucose into the cells. And group 2 usually have high Km or low affinity for glucose. So that means so that's why group 2 is kind of glucose dependent. When only we have high levels of glucose, then group 2 can be active and transport the glucose into like liver or beta cell, right? Pancreas. And group 4 is low Km and high affinities. So that means transporter. So group 4 is different because group 4 we know that depend on insulin, right? If we have a lower insulin, and the majority of group 4 is located inside of cell, not on the cell surface. So group 4 is kind of different. So depend on insulin. All right. So just wanted to mention you again that group 2, group 4, the regulated differently by group 2. Regulated by glucose levels, and group 4 regulated by insulin, right? Okay, so regulation. Let's take about 10 minutes break right now.
Oh my gosh, yesterday was like, but like our downstairs people were like, can you get the sunshine shop? Because there's lots of the pots or whatever and what? Yeah, that's what we saw. So like our landlord and we like that's what got yeah, just anything and
Oh, really?
and so we did, but like none of the guys were like, we sorted it all that, and we, I don't know where they went, like party and living out here.
Oh my god.
So I was like, well, just that's cold. So today I just went to the gym really early to pee.
Oh my gosh, really? Yeah.
I know, I think we just fixed it. Yeah, I think it's fixed. We need to
Obviously. I figured.
Restore the garbage that goes into the garbage can or it's like black. It's like those lights, like there's rectangle ones. So they have a whole light fixture next to the garbage and like so I watched doing that downstairs and figured out something.
Yeah, I did not get it. Did you? I made it up. I mean, all that work that we got, like, it all realized it. Like, Shane's list is just like 8. So what's the all you literally just said bring PS of your problem in your life. So problem, oh, what's my PE? Is it a personal airline? Slide where am I finding PS on the slide? Step 1, problem. Not eating, drinking. Step 2, epidemiology. My work, school life, and how it's changed. And then your signs and symptoms. Annoyed, feeling, angry, mad,
You can do whatever you want.
You literally can do whatever you want. That shit just wants you writing. Just go.
What?
You get on the list, bro. Really got money.
I want to fill the water.
Oh, you definitely gonna be late now. What'd you make up? Julia got it. I know. I'll get dropped at people. Which is Thursday. I'm sure she'll pass it. Or you just have after class, probably. I don't know what we're doing.
I literally took a bite and it was like the size of this. It was that big and I was dying. I don't remember. Because I was like, oh, one of these is hot. I forgot to remember which one. And it was so
They just want to go around with some who have some of their names throughout the year, and they want to see
Okay, everybody. Now let's talk about the regulation, the second levels called substrate cycles. Right? We just finished the regulation at the transport level, right? And substrate, and I just wanted you to try to remember that the three key reactions, right, in the glycolysis, and these are the three key enzymes: glucokinase, fructose kinase, and pyruvate kinase. Remember, there's three key reactions in the glycolysis pathway, right? And then also actually four key enzymes in gluconeogenesis pathway: PepCK, pyruvate carboxylase, and fructose 1,6 bisphosphatase, and glucose 6 phosphatase. And again, I mentioned that the key enzyme, why it's so important, because this enzyme are the place that hormone are the targets of the hormonal regulation, say, right? The target hormone regulation, and once we control this key enzyme activity, then we can control the whole pathway activities, right? So the key enzyme, again, is very important. And this slide just put the all glycolysis and gluconeogenesis pathway together. On the left side, from top to the bottom, is the glycolytic pathway, right? See, from the glucose can be metabolized to pyruvate, right? From top to bottom, the left side, it's glycolysis, and the right side from bottom to the top, started from pyruvate as a gluconeogenic precursors and to make glucose on the top, and it's the gluconeogenic pathway, right? And the key reactions in the glycolytic pathway, from glucose to glucose 6-phosphatase. So glucokinase is one of the key reactions, right, in the glycolysis, and the second one is phosphofructose 1 kinase. The key reaction in glycolysis catalyzes the conversion fructose 6-phosphate to fructose 1,6 bisphosphate. And the third, the key reaction is the pyruvate kinase, converts the phosphoenolpyruvate to pyruvate, right? And similarly, in four key enzymes in the gluconeogenesis, like pyruvate carboxylase, the PepCK here, and then fructose 1,6 bisphosphatase, and glucose 6-phosphatase, right? The four reactions, four key enzymes here. So I think this is a pretty good summary of the the regulation of the two pathways by substrate and hormones. For example, the fructose 6 phosphate and fructose 1 phosphate are the substrate, right? And these two different substrates can regulate glucokinase differently. For example, fructose 1 phosphate stimulates glucokinase, so activate glucokinase, right? So can increase the glycolysis activities. And fructose 6 phosphate basically inhibits glucokinase, so reduce or slow down the glycolysis, right? And insulin basically stimulates the glucokinase and so increase the glycolysis. And insulin does the opposite thing to the gluconeogenesis, right? So insulin basically inhibits the glucose 6-phosphatase, right? So slow down gluconeogenesis, which makes sense, right? During the fat condition, when we have a lot of glucose uptake from absorbed it from the diet, and we stimulate insulin, right? We try to utilize glucose and not to make glucose, right, in the fat condition. So insulin levels increase in response to the high blood glucose level, can promote or stimulate like glycolysis, but inhibits gluconeogenesis, right? So insulin does like two things to regulate blood glucose level here in these two different pathways. And similarly, you can see insulin right here. Insulin always stimulate the key enzyme in the glycolytic pathway. Insulin can also stimulate second key enzyme reaction, fructose, phosphofructose 1 kinase, and insulin can also stimulate pyruvate kinase, right? So all work together to stimulate glycolysis, but insulin actually inhibits, second key enzyme in the gluconeogenesis, inhibits fructose 1,6 bisphosphatase, and in this inhibition, insulin actually does not directly regulate this enzyme, right? Indirectly. So insulin first of all regulates fructose 2,6 bisphosphate. This is very important substrate, right? We're going to talk about it. And this substrate is not directly involved in gluconeogenesis or glycolysis, but independently. But this substrate is very important, regulates the the activity of the key enzyme in gluconeogenesis and also glycolysis in this fructose 2,6 bisphosphate, again the key substrate, right? And insulin definitely also inhibits the PEPCK activity. And it also have some other substrate, for example citrate, and let me see what it does. So AMP, the substrate can also regulate the different enzymes. So you probably you don't need to remember all the different substrate how they regulate the enzyme activity, but I just wanted to focusing on a couple of them, right? We're going to talk about the later. Okay, so this just give you a overview in how nutrient and hormonal regulation of there's two different pathways of glucometabolism. So we know that nutrients, physiological stimuli can regulate the hormone release. For example, blood glucose level, right? It's a good example. It can increase the insulin levels. So once in the fat condition, so once insulin is released, and insulin can regulate the enzyme activity, can regulate the transporters, right, and group 4, and also can regulate the key enzyme activities in glycolysis and gluconeogenesis pathway. And it also can hormones can regulate the gene expression. That means of the enzymes, just directly regulates the levels, the production of the enzymes. So activity and then production can be a different concept, right? So for the activity regulation, basically is a short-term regulation. For example, we're going to talk about later like phosphorylation. So you can turn on and off the certain key enzyme, that can happen very quick, like couple minutes, right? And the long-term regulation, for example, regulate gene expression, that takes a longer time, maybe hours or days, or So protein phosphorylation. So protein phosphorylation means that the phosphate group can be added to the protein amino acid residues on certain proteins. So very common amino acid that can be added phosphate group are serine, threonine, and tyrosine residues. right? It is a reversible process and is a very important regulative mechanism. For example, some of the enzymes, once the phosphate group is added, can activate enzyme, or if phosphate group is removed, you can inactivate the enzyme, right? So it's sort of the regulative mechanism to, regulate enzyme activities. Actually, two different type of enzymes that catalyze the phosphorylation. For example, kinase normally catalyze the addition of a phosphate group to the amino acid residues of a protein or enzyme. And so kinase basically regulates the phosphorylation of the enzyme or protein or enzyme, right? And phosphatase catalyzes the remove of the phosphate group from a protein. So, and this process called dephosphorylation, right? So phosphatase catalyzes the dephosphorylation, while kinase regulates phosphorylation of a protein or enzymes, right? And phosphorylation, when phosphate group is added to the protein and can causing the conformation change of protein, so then leading to the change in enzyme activity, right? So it's sort of a mechanism. So many of the enzymes and transporters that can be phosphorylated or dephosphorylated, or can be switched on and off, right, by adding phosphate group or remove phosphate group from the enzymes. It's just a mechanism, right, for regulating the enzyme activities. The one of the actual mechanism. There are actually multiple different mechanisms that protein activity can be regulated, but most common, actually, the phosphorylation is the most common mechanism, right, for regulation of enzyme activities. Okay, so hormone regulation. So let's talk about insulin. So in which physiological condition insulin levels rise? Do you guys want to fill in the blank? In which physiological condition, fat or fasting condition? Which condition insulin level should be increased? Fat condition, right? Yes, so under the order called a postprandial or fat condition, right, insulin levels rise. And how about glycolysis and the glycogen storage activities? How insulin regulates the glycolysis and glycogen storage? Increase or decrease? Increase, right? So insulin basically increases the glycolysis to allow the more glucose be metabolized, right, from the glucose from the diet, and also wanted to store the glucose in the glycogen, right, in the form of glycogen in the liver during the fat condition. And what about the gluconeogenesis or glycogenolysis? Glycogenolysis, it means glycogen breakdown, right? So how about the gluconeogenesis and glycogen breakdown pathway? How insulin regulates? What do you think insulin should regulate this to gluconeogenesis and glycogen breakdown? Decreases, right? So we should decrease, right? We don't need make glucose or break down stored energy, right? We have enough energy from diet. And what do you expect the expression of the glucokinase gene, which is a key enzyme in glycolysis, right? So how would insulin regulate this? We know that insulin regulates the gluconeogenic pathway, right? So what do you think of how insulin would regulate the a key enzyme in glycolysis. Could increase, right? So activates or increase the expression of glucokinase gene. And what about the PEPCK gene? PEPCK is the key enzyme in the gluconeogenesis, right? So how would insulin decrease, right? So insulin inhibits or decrease, we use the different terms, inhibits expression of PEPCK gene expression, and stimulates glucose transporter into which tissues? Which tissues, glucose uptake regulated by insulin? And which transporters? We say group 4, right? And muscle adipose tissue, yes. So muscle adipose tissue, so insulin stimulates group 4 translocation to the cell surface, and glucose transporter into skeletal muscle and adipose tissue, right? So what about the net results? Blood glucose level when insulin level is high. Decrease, right? Yeah, it's a decrease. Yeah, great. So you just try thinking how insulin coordinately regulates these two different, so opposite, glucose metabolic pathways in the fat condition by insulin. And okay, how about the counter-regulatory hormones to insulin action? For example, glucagon, epinephrine, glucocorticoid. These hormones are usually elevated during the fasting, right? And their action is opposite to insulin. So in which physiological condition we expect the levels of these hormones will be increased? In which physiological? Fat or fasting? Fasting, yes. So fasting or starvation, the glucagon, epinephrine level or cortical level will be increased. And which tissue is the main target tissue of a glucagon? What glucagon does, do you guys know? In general, glucagon is raise blood glucose level, right? So what glucagon does is to raise blood glucose level during the fasting. So it's just opposite to insulin. Insulin reduces the glucose level, right? So which tissue, thinking about, what kind of pathway that can be activated and to raise the blood glucose level? And then thinking about which tissue does this, metabolic activity. Thinking we have 5 different pathways. Thinking about which glucose metabolic pathway active then can increase the blood glucose level. We just talk about one, right? Gluconeogenesis. Yeah, liver. So liver is the main targeting tissue of glucagon. So glucagon can act on raising blood glucose level in the liver. So which pathways in the liver that can be activated during the fasting by glucagon? Gluconeogenesis, yeah, gluconeogenesis definitely, and also very important. The pathway that need to be activated during the fasting. The glycogen breakdown, right? So glycogen breakdown, we need to break glycogen to release glucose as well. So actually two important pathways that contribute to the blood glucose during the fasting: glycogen breakdown and gluconeogenesis, right? So the liver is the main target of this counter-regulative hormones, and the two pathways can be increased by this counter-regulative hormones, right? And again, gluconeogenesis and glycogen breakdown. So what do you think? Glucagon should change the expression of PepCK genes. PepCK is again the key enzyme, right, in the gluconeogenesis. Increase, yes. So glucagon will expect to increase expression of PepCK gene and stimulate glucose output from the liver, right? Again, through the two different pathways, glycogen breakdown and gluconeogenesis. And net result, increase, right? Try to raise the blood glucose level during the fasting. Okay. All right. So the next couple of slides, I think I'll just list some questions that help you to study, what you need to know about, glucose transporters and hormonal regulations like, insulin, glucagon, and other counter-regulatory hormones, how they regulate the glucose metabolism. Just connect like a whole body, right? I mean, as this, how they regulate this glucose metabolic pathways to maintain the blood glucose level in fasting conditions, right? And substrate cycles. I listed here the substrate. All the substrate that you guys need to remember, right? How they regulate the glycolysis and the gluconeogenesis, and maybe later the glycogen, synthesis of glycogen breakdown pathways. Okay, so I think we have some sort of exercise here. So let's do this choose both selections. In terms of glycolysis and TC cycles, indicate whether the following statement about the glycolysis is true or false. All right. So first question: glucose is oxidized to either lactate or pyruvate via glycolysis. It's true, right? Obviously, yeah. And second one, oxygen is not required for glycolysis. True, yes. 3, all cells can do glycolysis for producing energy from glucose. True? Well, why you shake the head? It's true, right? Every type of cells is able to utilize glucose. Okay. Number 4. So glycolysis is the first step of a glucose metabolism. It breaks down glucose and generates intermediates, which can be further metabolized or converted to intermediates of other pathways. For instance, lipogenesis. Remember the TC cycles? Yes, right? Glucose can be converted to fat, right, through the TC cycle. Citrate, right? Citrate is the key molecules. 5. The rate-limiting steps of glycolysis is a glucokinase catalyzed phosphorylation of glucose to glucose 6-phosphate. Right? Yes. The first key reaction, remember, right? Okay, so now the second slide. Okay, so the first question, the TCA cycle generates ATPs. Yes, right? And second, the TCA cycle is common to oxidation of carb, fatty acids, and proteins. Yes. Pyruvate is the starting molecules of TCA cycle. Pyruvate? No. Which molecules is it? Acetyl-CoA. Yes, great. Thank you. Okay, number 4. TCA cycle is the only cycle or pathway that produces reducing equivalent. It's the only cycle pathway producing reducing equivalent. Is that true? In terms of glucose metabolism, which other pathway that can produce ATP, reduce equivalent? Glycolysis, right? Remember. So it's not true. And 5. TCA cycle is the ending oxidative pathway for all 3 micronutrients. I know some of you think yes, some of you think no. Well, depends. I mean, if we're thinking just the metabolism, like a carbon metabolism, this is yes, right? I mean, we break down the carbon to CO2, right, water. And if we're thinking the ATP production, well, actually, NADH or FADH will need further to the electron transport chain, right, for ATP production. So it's not. So probably it's not very good question, but it's good thinking, right? Okay. I don't know. We'll see. Yes or no. 6. So the first reaction in TCA cycle is the condensation of oxaloacetate and acetyl-CoA forming citrate. Yes, right? Okay. All right. Oh, 2 more. So, so the end product of TCA cycle are CO2. Ah, too easy. Yes, of course. And second, oxaloacetate and citrate are the intermediate that can be removed from TCA cycle for gluconeogenesis and lipogenesis respectively, let's say. Right? Yes. Okay. All right. So I think that's all for this section. Okay. Okay, I think we kind of a little bit behind, right? Let me just try to see what we need to. Okay. Let's just quickly go through some. Maybe it'll take 5 minutes, right, to go through some of the, slides. So I think the next step that what we're going to do is to go through the specific reactions, right, and specific key reactions, enzymes, and how this reaction key enzymes are regulated by hormone and substrate. So if you still remember, this is again the first key reactions, right, from glucose to glucose 6-phosphate in the glycolysis, and the last reversal reactions, right, in the gluconeogenesis, right. So this reaction kind of paired, in the 2 different pathways, and this reaction, the different direction catalyzed by different enzymes. Again, this is irreversible. The reaction is irreversible, so require the different enzyme to catalyze, reaction through the different direction. And let's talk about glucokinase or hexokinase. So I mentioned before, the glucokinase basically is predominant in the liver, right? A hexokinase in the muscle. So the catalyze the same reaction, but the different tissue distribution. And hexokinase have a lower Km, a high affinity for glucose, and this enzymes, hexokinase, is usually coordinated with group 4, means that the because the group 4 is the predominant glucose in the skeletal muscle, right? So glucose is transported by group 4 into the muscle cells and then catalyzed by hexokinase to phosphorylate glucose to glucose 6-phosphate, right? So this process is called, coordinate with the group 4, which makes sense, right? And this hexokinase usually allosterically inhibits by its product glucose 6-phosphate. So glucose 6-phosphate serve as the substrate that can regulate the hexokinase activities, right? So hexokinase activity can also control the glucose uptake, and phosphorylation basically is the first reaction. And glucokinase again is liver and pancreas, right? More predominant in the liver and pancreas, and have a high Km and low affinity for glucose, and usually coupled with group 2, which also makes sense, right? Liver and beta cell usually have a higher group 2, so group 2 activity is coordinated with glucokinase. And this enzyme is different from hexokinase. It's not regulated by glucose 6-phosphate, but it's actually regulated by fructose 6-phosphate. Right? So this enzyme in the different tissues regulated by different substrate. And let's just go to the next slide. So this slide just shows us how glucokinase is regulated by hormone and substrate. It's a pretty busy slide, right? This represents the liver cells, and this is the nucleus, and this is cytosol. And we know that glucokinase activity, or the first reaction, the glycolysis, occur in the cytosol, right? So this all the glycolysis pathways. So glucose, first of all, is transported by group 2 into the liver cells, and glucose can be phosphorylated glucose 6-phosphate by glucokinase, right? And glucose 6-phosphate can be converted to fructose 6-phosphate, right? And of course can also be further metabolized, right, through the glycolysis pathway. And insulin is an important regulator of glucokinase, so basically stimulate, right, increase the glucokinase activity in the fat condition when insulin level is high. And glucokinase, this enzyme is kind of interesting. This enzyme usually is located in the nucleus. So when it's located in the nucleus, it's impossible for it to function, right? Because this reaction actually occurs in the cytosol. So in order for glucokinase to be active, the glucokinase need to be transported from nucleus to the cytosol, right, to the place where the reaction occurs. But in the sort of rest condition or the basal condition, the glucokinase is bound to called a glucokinase regulatory protein. So the glucokinase regular protein sequesters the glucokinase in the nucleus, so wouldn't allow the glucokinase release into the cytosol. So under the the basal condition, and there is actually two important substrates that can regulate the dissociation of glucokinase from glucokinase regular protein. So allow the glucokinase release from nucleus into the cytosol for its activities. All right. So, for example, fructose 6-phosphate is a substrate that can inhibit this dissociation. right? So that means when cells have a higher fructose 6-phosphate, the glucokinase will be sequestered in the nucleus. And the fructose 1-phosphate, this is a substrate that metabolizes from fructose, right? So fructose 1-phosphate can promote the release of the glucokinase from the nucleus to the cytosol, so then can increase or stimulate glucokinase activity. So this is sort of the mechanism, how glucokinase can be activated by this this substrate fructose 1-phosphate. So what you try to remember is the three substrate. Three substrate. So glucose can also promote the release of the glucokinase, right? So 2 substrate, glucose and fructose 1-phosphate. These 2 substrate can stimulate release of glucokinase from nucleus to the cytosol and to activate the glucokinase, right? And 1 substrate, fructose 6-phosphate, actually inhibits this association. So still wouldn't allow the glucose kinase released from the nucleus. So then inhibits the glucokinase activity, right? Does that make sense? So try to remember these 3 substrates and how this 3 substrate regulates the glucokinase release from nucleus and activates the glucokinase. And for hormones, that insulin actually directly regulates the glucokinase activities, right? So I just want to stop here for today, and we have informal writing, right? Let's see. Okay, so this will be the question for today's informal writing assignment. During the starvation, how does insulin levels change, and how does insulin regulate glycolysis and gluconeogenesis in the liver? Right? Oh, I just wanted to remind you that I'm already posted the homework assignment 1 on the Moodles. And so again, later I will also post the homework assignment 2, but you only need to select 1, right? You only need to complete 1. All right. Excuse me.